Okay, let's get started. We'll jump right into it. So this is the Biology Paper 2 session. So just like all the other classes, I'll be going through some exam questions and content at the same time. It's not everything, but it is majority of things that potentially might come up in tomorrow's exam. And like always, if you've got any questions, just drop them in the chat as I'm going along and I can answer them as we're going. So let's get right into it. So define the term homeostasis for 2 marks, tends to come up almost every single year. And the 2 points that they're wanting is, for the first mark, they want you to say the regulation of internal conditions. And the second mark is explaining why do we exactly need regulation of internal conditions. And it's for optimum selectivity. So we want our bodies to basically be at the most optimum condition they can be. So for 2 marks, make sure you state them 2 points clearly as well. Another question that they can ask you about homeostasis as well is, well, what do we exactly need to control? What internal conditions do we need to regulate? Temperature is a big one. Water levels and glucose concentrations. These tend to be ones that be on the mark scheme. There's other stuffs as well, like pH, ion concentration and such, but they're the 3 main ones: temperature, water levels, and glucose concentration. And the 2 ways that you need to know for GCSEs is the nervous system and the endocrine system. So let's go through it. Let's go through the nervous system first. Make sure you know the differences between each as well. So the nervous system, this is working with electrical impulses, and try and not use the word nerves. Try and use the word neurons instead. Neurons really are just nerves, to be fair. And then for the endocrine system, this works with the glands secreting hormones. And hormones are just chemical messengers. So we'll compare both of them, but let's just go through each of them quickly on what you need to know about them. So for the endocrine system, you need to know where each gland is, and you need to know what hormone they produce. Now, remember, the hormone doesn't actually carry out the job. The hormone is just a chemical messenger. It's telling your body to do something. So as an example, let's look at the pancreas first. The pancreas produces insulin, and insulin regulates blood glucose concentration. So it's not actually changing the blood glucose concentration, it's regulating it. For higher, you need to know about glucagon as well. So I'll cover that towards the end. So that's the pancreas. It produces insulin and glucagon for controlling the blood glucose concentration. You've got the pituitary gland, which is not part of your brain, but it's just situated at the base of the brain. And we call this the master gland, so it tells the other glands what to do. So for higher students, it's quite important for negative feedback, like with the thyroid gland with thyroxine and TSH and so on. But for both foundation and higher, just make sure you know the pituitary gland is the master gland. It tells other glands what to release. We've then got the thyroid gland in our neck, and that produces thyroxine, the hormone thyroxine. And what this is responsible for is controlling our metabolism. So just remember, our metabolism, that's just the sum of all the chemical reactions happening in our body. Okay, so that's for the metabolism. We've got the adrenal glands. These produce adrenaline. What does the hormone adrenaline do? It's the flight or fight response. So a good way to remember it is, just imagine you're on a roller coaster. You get that adrenaline rush when you go down a roller coaster. That's adrenaline getting released in your body. So it's making you scared in order to protect yourself. So that's the adrenaline effect. And then if you are male, you've got testes, which produce testosterone, which form after puberty. So that's the reason why males get deeper voices, they get facial hair and so on. And then for females, they've got the ovaries, which produce estrogen, but they also produce progesterone as well. So make sure you know them 2 key differences as well. So just be aware, with all these hormones, these all produce quite slow responses. Like it's not quick, like a nervous response, which we'll come on to. So it's quite slow, it's quite generalized, and it's not really specific as well. Moving over to the nervous system, make sure you know the order. So the stimulus is what you're detecting. So say, for example, you put your hand under hot water, for example. The hot water is the stimulus. The receptor is something that's going to detect the stimulus. So for example, skin, you have receptors in your skin which would detect that hot water, which essentially you want to pull away from. So that's your receptor. It then follows this path of neurons, which in other words are just nerves. So your sensory neuron, your relay neuron, and then your motor neuron. And this part, these parts are all in part of the brain and the spinal cord. So that's why it says here inside the coordinator. You need to know how the neurons actually join together, and they join together by a gap called the synapse. So I'll just show you on, let me just get this set up. Okay, so this is the gap here. So each neuron, they're not necessarily touching each other. There's a gap in between both of them, and that gap's known as the synapse. And between that synapse, there's chemicals, which are called neurotransmitters, which pass through each of them neurons to be able to pass through that electrical. So that's how they're connected. Okay, so make sure you know the order. After it passes through all of them, that then gives the effect to the effector. So in the example we just spoke about, your hand running under really hot water, you're pulling it away. Your effector is your muscle contracting because you're pulling your hand away. So that's your effector, and then the response is you've moved your hand. Now notice with the nervous system, the effects are really fast. They have to be fast. If you are burning yourself or you, I don't know, stabbed yourself with a pin or something, you need it to be quick. You need to respond to it quick. And that's the main differences with nerves and hormones. So you can see with the nerves, should really say neurons instead. It's faster action. That's the reason why it uses electrical impulses. Hormones are quite slow. So for example, if your blood glucose concentration drops, it's not going to straight away increase. It's a slow process. Nerves act for a very short time. So once you've pulled your hand away, that's it. You've pulled your hand away. The response is over. And it acts on a very precise area as well, compared to hormones, which act more in a general way. If you're releasing a hormone, it's not going just in one particular area. It's just very general. So for example, when you get the adrenaline rush, you don't just feel it like in a particular area, your whole body tends to feel that adrenaline rush. So that's the effects of hormones. Let's talk a little bit about glucose concentration. So what's the importance with blood glucose concentration? Remember, we use glucose in respiration. So I'm just going to do this down here. So we need glucose for respiration. Make sure you know the equation for respiration and photosynthesis as well for this exam. So for respiration, we breathe in, I'll just try that, we breathe in oxygen, which reacts to our glucose, which is in the blood, which is why we're really specific to saying blood glucose concentration. That then releases energy. The whole point of this is for energy. And we breathe out carbon dioxide, and we also release water. So this is the key thing that glucose is needed for. Now, if your blood glucose concentration throughout the day is always going up and down, so this is where the idea of negative feedback comes into. So what is negative feedback? Well, let's say this is the time of day. So as you're going along the x-axis, your glucose concentration is never just one level. It's always bouncing up and down, up and down, up and down. So we need a mechanism in the body which, whenever it gets too high, something in the body needs to bring it down. But then when it's gone low, something needs to bring it high again. Now it's come high, it needs to come low again. So this process is known as negative feedback. Now, for both foundation and higher, you need to know about blood glucose concentration when it's low and high. So blood glucose concentration. If your blood glucose concentration is too high, so we're on top of the peak, we need to bring it low. So what happens is, make sure you remember these steps. And when you're talking about glucose concentration, please make sure you say blood, otherwise you will lose the marks as well. So if your blood glucose concentration is too high, your pancreas is going to release insulin. So remember, insulin's the hormone that's just getting released. You can think of hormones as, well, they're chemical messengers. Insulin's going to tell your body to do something. It's not actually going to do anything. So the pancreas has released insulin. What's this going to cause? The blood glucose will get moved to cells. And the reason why it's moving to the cells is to be used for respiration. So that's what the insulin is telling your body to do. So it's saying that glucose that's in the blood, move it to the cells and go use it for respiration, because then that means you're going to lower your blood glucose concentration. But what if you don't need it for respiration? What if you don't need that energy at the time? Maybe you might be sleeping and your pancreas is releasing insulin to lower your blood glucose concentration. So what will happen at that point? Any excess glucose is stored as glycogen. So just be careful of these words, glucose and glycogen, don't get them confused. Glucose is just small molecules. Glycogen is loads of glucose combined together. So any excess glucose or spare is stored as glycogen in the liver and muscle cells, because it wants to get it out. Liver and muscle cells, it wants to get it out of the blood, because the whole purpose of insulin is to lower the blood glucose concentration. So now we've lowered it. So now we're at this point here. So we started here, it was too high, the pancreas released insulin and it decreased it by using the glucose for respiration, and any excess glucose was stored away in the liver and muscle cells. But now the concentration's gone too low, so it needs to bring it back high again. So what happens when it's low? Now this is just for higher tier only, so foundation, you don't need to know this. If it's too low, this time your pancreas is going to release glucagon, which is a different hormone. So just want to emphasize, please don't get confused with glucose, glycogen, and glucagon. You don't want to lose marks just because you've wrote the wrong thing. Now what happens is when the pancreas releases glucagon, it tells this excess glucose here that was stored in the liver and muscle cells as glycogen, basically, to change back to glucose and go back to the blood basically. So release back to the blood. So if you've got that glycogen, you've broken it back down to glucose, you've now increased the concentration of your glucose again, but now it's too high. So then your body needs to go back down again. So it's going to do this process again. So it's going back and forth between each of these two processes. So that's why it's called negative feedback. So this tends to come up almost every single year as well in the paper. So make sure you've got a good idea what happens when your blood glucose concentration is high and low. So let's go back up to the exam questions. If you've got any questions as well, as we're going along, do drop them in the chat. Okay, so what I've just spoken about there is highlighted here as well. I'll get these notes sent out after as well. So this is just what I've spoken about on the bottom. So your marks come from the gland that you talk about, pancreas. Say what it releases, the hormone, and then what does that hormone cause, basically. Okay, so let's look at this past exam question. It says here, person A and person B had a test to measure the concentration of insulin in their blood when they were fasting. So it's concentration of insulin they're looking at, so the actual hormone itself. So keep in mind, insulin gets released when your blood glucose concentration is too high. Okay, so keep that in mind. Table 4 shows their results. So we can see with person A, they've got a very high blood insulin level. So it was quite, they tricked you really. Usually they ask you questions about blood glucose, but they're saying blood insulin here. So you can see here, person A has got quite a high concentration of insulin. So they're producing a lot of insulin. And then person B, they've got a low insulin amount. So always understand the data that you're given the questions tomorrow rather than just jumping straight into the question. They've also told us what a normal range is. So a normal range is between 50 and 175. So we can see person B is not a normal amount of insulin, but person A's got too high insulin. They're higher than 175. So this is too high and then that's too low. Okay. So both of them basically have diabetes, person A and person B. Suggest which type of diabetes person A and person B have. So we can see they're both abnormal. This is too high and this is too low. Remember, type 1 diabetes is where your body is not producing enough insulin or any insulin. So looking at this, we can say person B is type 1 diabetes. And the reason why is because they're not producing enough insulin. So we say this is more of a genetic condition, type 1, that your body is just not producing enough insulin. And we can see that because they're producing 20 units. Person A has way too much insulin in their blood. So then that means their body is not responding to the insulin, and that's type 2 diabetes. So they've got loads of insulin, they're not responding to it. And we say type 2 diabetes is caused because of lifestyle. So the reason we would give is that 280 is too high. So the body is not responding to the insulin. So if I just quickly show you the comparisons between type 1 and type 2, if you look here. So diabetes is basically where you cannot control your blood glucose levels. I have wrote sugar level there, but you should say glucose levels. The reason why that's bad is if it's too high, you can't bring it down again, and that's the issue. So you can see with type 1, the pancreas cannot produce enough insulin, and type 2 is where your cells no longer respond to the insulin produced. So in that exam question, the insulin was way too high because the body's not responding to it. Okay, let's go back to the exam questions. Okay, so that's a bit about the endocrine system, nervous system, and so on. There is more to it, but I've picked out quite a few there. Let's move over to quadrats. So what is a quadrat? It's one of those square things that you throw in a field to, and then you basically count how much are in there. So in this case, let's look at this exam question. Some students estimated the population of daisy plants in a field. So estimated the population because they're not going to a field and they're not counting every single daisy plant that's in that field. They're going to be using the quadrats, throwing them, and then maybe comparing it against the whole field size. So this is the method used. Place a quadrat randomly on the field. Count and record the number of daisy plants in that quadrat, and then repeat steps 1 and 2 another 4 times. How come the students have made sure the quadrats were placed randomly? Or how could they, sorry. So the most common answer you could just say is a random number generator. So meaning that you'd label like each part of the field, assign a number, for example, and then use a random number generator just to randomly pick one of those. So there's no bias, because if you were looking at the field, you might have some sort of bias and choose the areas which have a lot of daisy plants, and that's not representative to the whole field. So yeah, so you could just say random number generator. Describe the piece of equipment called a quadrat. You could just say it's a square grid, because that's what it is, right? It's just one of those square grids that you're just throwing down. Okay, so the students noticed that the daisy plants growing near a building were smaller. Explain why smaller daisy plants grew near the building. So in the context of this question, why would daisy plants be smaller near a building? What is happening near the building? Well, if there's plants near a building, that means they're going to get less light if they're near a building. And if they're getting less light, what's light needed for? Light's needed for photosynthesis. So therefore, less photosynthesis happening. So plants were smaller. So you will get a lot of questions like this where it's one of those where they want you to figure out what's actually happening. So the fact that they're near a building, what's lacking near a building, it's light, and what's light needed for photosynthesis. So they're your two main marks where they come from. Okay, so you might also get questions to do with abiotic and biotic factors. Remember, abiotic are non-living factors which are affecting an environment, and biotic factors are living factors which affect an environment. So just make sure you know this list because you don't know how much they could ask you tomorrow. So abiotic factors are stuff like light intensity. Why is light intensity a factor? Just like that last question. Because there was less light near a building, the plants were not growing as well as they could. Temperature, again, you need a good temperature for photosynthesis to take place. Moisture levels, which is the amount of H2O in the atmosphere, so that can contribute to stuff like transpiration and stuff. If we look at the biotic factors, so new predators arriving, which is a living thing. If a predator came and they took over some sort of land, they would kill all the plants there, for example. New pathogens, remember pathogens are actually living, and if diseases spread across a field, it could kill plants, for example, or so on. So yeah, just make sure you know the differences and a few examples of abiotic and biotic factors. Okay, key definition. So biodiversity is the variety of all different species found in an ecosystem. So that's quite a common question where they could just ask you what's the definition of biodiversity. So it's a variety of all the different species found in an ecosystem. So if you want to keep an example in mind to help you remember it, so the Amazon rainforest would have a very high biodiversity because there would be more different species compared to somewhere like in the city where we all live. So this would have a high biodiversity. Okay, what's a genome? A genome's the entire genetic material of an organism. You might get asked about the Human Genome Project as well. That's quite a common question. And they usually ask, what's the advantages of them doing the Human Genome Project? It allows us, it allows the field of medicine. Essentially, we can screen for diseases earlier, so like inherited disorders and stuff. We can figure it out and make medicine basically a better field. So that's how the Human Genome Project contributed. Okay, we've got another question here. Why does deforestation take place? I pulled this from the mark scheme because it comes up quite often and it's the same thing. So deforestation, remember, that's the process where trees get chopped down. We'll just call it trees chopped down. And the reason why they do it, usually it's for space, basically, but the mark scheme wants a bit more than that. They want you to explain how exactly is that space used. So the space could be used to grow crops. So for example, farmers, they need a lot of empty land to be able to grow crops. So that's why deforestation takes place. To raise cows, again, it's a farming reason, and to build. So for example, like cities, we need space, we need to be able to get all the trees down. So yeah, here's a few others that they would allow, but I would recommend just sticking with these three reasons when they ask why does deforestation take place? If the question says just state one thing, please just only state one thing. Don't just write multiple things, because even though they all might be right, you still would lose marks because you've not answered the question by just listing one thing. Another common question is what causes extinction? So remember, extinction is when a species is basically finished. It no longer exists. So anyone from a new disease, because that disease could essentially wipe out all the species. This is why it's really important for things to have genetic variation, because if everything was exactly the same, so for example, when animals are being breeded, you want to keep one characteristic. If you keep that one characteristic and that characteristic could be contributing to like a disease, then that disease would eliminate all of them because there's no genetic variation. So that's why genetic variation is also really important as well. So what causes extinction? A new disease, a new herbivore, climate change, global warming. So for example, like in the Arctic, polar bears, penguins and such, they need that cold climate, and global warming is damaging their ice caps, so that could cause extinction. Volcanoes, earthquakes, tsunami, meteors, that's self-explanatory. A new competitor or lack of pollinators. So lack of pollinators mean that the producer right at the start of the food chain is not essentially going to be growing. And if the start of the food chain is not there, how's the rest of the food chain going to be there? That's what it's referring to. Ignore new predator, allow drought, flooding, allow plant collectors. Yeah, so just stick with these ones. Okay, we've got another exam question here. So the cat breeder wants to use selective breeding so that all new kittens have blue tail tips. So selective breeding basically means they're getting a certain characteristic and they're repeating that over and over and over again. So describe the process of selective breeding the cat breeder could use. Again, this is particular to cats, but it can be used for anything. They could ask you a selective breeding question on anything, and it's still the same format always used in every mark scheme. So choose the cats with the blue tail. So your first part, you always say choose the desired characteristic. So if you get I don't know, a question tomorrow on like you want I don't know, a particular color eye on a dog, for example, that's the desired characteristic you're choosing. So that's where your first mark comes from. Your second mark comes from breeding the ones that have the desired characteristics together. So you breed them cats together. Choose offspring with the blue tails and breed them together. So you're basically repeating the process. So when you get your offspring, they'll have the desired characteristics, and then you keep breeding them over and over again. So yeah, that's where your final mark comes from, just repeating until they all have the blue tails, basically. So that's selective breeding, and it's always in that same format. It's just a different example that they give you in every exam. Okay, moving on. Okay, so with each pregnancy, the cat breeder expected that 50% of the kittens would be male, 50% would be female. The sex chromosomes in cats are inherited in the same way as humans. So this is quite important. In humans, you know that male is XY and female is XX. So that's the reason why they told you that sentence. It's told you the chromosomes are X and Y, give the combination of the chromosomes present in male and female. So even though it's talking about cats, it's the same thing you've learnt. So male being XY, females being XX. The cat breeder expected 50% male and 50% female kittens. Complete the Punnett square. The female cat would be XX, the male would be XY, and we're just crossing these together. So these Punnett squares, you need to know them for any disorders as well, so like cystic fibrosis, polydactyl, which we'll go over as well. So this first one gives us XX, this one here gives us XX, this one here gives us XY. Make sure you put them in alphabetical order, so X before Y. And then on the bottom here, we've got XY. So we can see it's 50% chance of XX and 50% chance of XY. So we've just proved it. So for 2 marks. Now I just want to show you another question like that. And they gave 5 marks for this one. So this was on a paper 2 years ago. Okay, let's look at this one. So it says here a person with genotype D, capital D, small d, and a person with genotype small d plan to have a child. Now, just before doing this question, just want to know what so alleles are basically different versions of the same gene. So different versions of the same gene. So for example, up here, it's dominant and recessive that we've got here. So 2 different alleles we could have. We could have dominant, we could have recessive. Now, dominant is represented by the capital letter, recessive is represented by the smaller letter. Now, for recessive, you need to have both small letters, and we represent every allele basically with 2. So, for example, dominant, it could be capital D, because one comes from your mum, one comes from your dad. Or we could have capital D, small d. So these are both dominant. With recessive, We need both of them to be lowercase, so not just one. So they're our combinations. We call this one here homozygous dominant. We call this one heterozygous because it's different. And we call this homozygous recessive. So homozygous is where both alleles are the same case of letter, so capital, capital, or lowercase, lowercase. And heterozygous is where it's different. And they're your dominant and recessive. Remember, a dominant disorder you need to know about is polydactyl, which is what I'm going to cover now. And a recessive disorder you need to know about is cystic fibrosis. Polydactyl is where you've got extra toes and extra fingers. Cystic fibrosis is where the lungs are producing a lot of mucus. It causes a lot of breathing problems. And these are all inherited disorders, so they're genetic, so they're non-communicable. You can't pass it on to somebody. Let's go back to this exam question. So a 5-marker. Okay, so a person with genotype capital D, lowercase d, and a person with genotype lowercase d, lowercase t. Remember, genotype is actual genes. Phenotype is what's physically shown because of them genes are expressed. Determine the probability that the child will have polydactyl, knowing that this is a dominant disorder, meaning you only need to have at least one capital letter. So you could have 2 capital letters or you could have 1 and they would get polydactyl. Identify any offspring genotype that would have polydactyl. Okay, so crossing this over, for this first box, we get capital D, lowercase d. We always put our capitals before the lowercase. This one here, small d, capital D, lowercase d, and then d, d, just like so. So we can see that this one will have polydactyl and this one will have polydactyl. So let's just write that here. Because it's dominant. So because it's 2 out of a possible 4 that has polydactyl, which is just a half, so it's just 50%. So they're quite easy, easy marks to get here. They'll never tell you for polydactyl and cystic fibrosis whether it's a recessive or dominant one, because they want you have to know that. But if they do give you a complete random disease, they will tell you whether it's a dominant or a recessive disease. Okay, I'm just going to quickly do an example on cystic fibrosis as well, just in case that comes up. So remember, cystic fibrosis is a recessive disorder. So basically meaning we need 2 lowercase letters. Now, let's say we've got 2 parents who are carriers. That's all they've told us in the question. Now, what they mean by carrier is they're carrying the gene, but they're not showing the gene. So in this case, I'll just call it capital C, lowercase c. This is a carrier. Because they've got 1 lowercase letter, they're carrying the gene, but the parents do not have cystic fibrosis because they need 2 lowercase letters. So let's say they said work out the probability that these 2 parents will have a child with cystic fibrosis. I'll just try CF. So because they've told us both parents are carriers, yeah, the Punnett squares are nice, easy 5 markers. If they give you something you've never seen before, it's still the same thing. They'll just tell you whether it's dominant and recessive and just follow it that way. So they've said 2 parents are carriers. So meaning the dad will have capital C, lowercase c, and the mom will also have capital C and lowercase c. If they ask you in the exam as well, like why do the parents not have cystic fibrosis? It's because they don't have, they're just carriers. They need 2 recessive ones to have cystic fibrosis. So for this first one here, we've got 2 capital C's. Here we've got a capital C, lowercase c. Here we've got a capital C, lowercase c, and here we've got lowercase c, lowercase c. So this is dominant, this is dominant, this is also dominant, and the only one that isn't dominant is this one here. So there's a 1 in 4 chance that the child will have cystic fibrosis. So you can just say that's 25%. So nice easy marks. You don't want to be losing marks from Punnett squares. Let's go back up. Okay, so a food chain. Remember with the food chain, we've always got the producer at the beginning. We've then got our consumers after them. So it says here which term describes a spider in this food chain. So it's not the producer, it's not the primary consumer because a black fly is a primary consumer. So it's the secondary consumer because it's the second one after. Okay, it's pretty straightforward. It says many of the spiders in the garden died. Okay, so this part of the food chain was getting affected. What is likely to happen to the number of black flies in the garden? Well, if there's no spiders to eat the black flies are not decreasing because there's no spiders to eat them. So therefore the black flies would increase. It says give a reason for your answer because there's no spiders to eat the black fly. Try and be really specific in your answers. Like, don't say to eat them, like what is them that you're referring to. You don't want to be losing easy marks like that. Okay, the question's continuing. Table 2 shows the estimated biomass of organisms in the garden. So looking at this, so bean plants, black flies, spiders, blackbirds, it's just going across the food chain. So the producer, then the consumers after, it's going in order. And looking at this table, I can see the numbers decreasing. So it starts off with a really high biomass and it's decreasing as you're going along the food chain. So what conclusion can be made about biomass in the food chain? You can say as you're going along a food chain, there is a decrease in biomass. That's why it's really good to critically analyze all the data you're given before you read the questions, because you might have already answered it yourself just by analyzing the data. It says here, explain why a garden is not a stable community. Okay, so in a stable community is one where the number of species will basically stay the same, so stable, nothing's coming in and out. But a garden, there's always organisms being added in or removed. So that basically means that it's not stable. So that's the point I've made. I need to explain it now. So you say, so the population sizes are not constant. So remember, the definition of a stable community is where the population sizes just stay constant and it just remains constant and it just stays in there. But for that to happen, nothing could come in and out of that community. So that's what that's referring to. Okay, so the ruler drop test. This might be one of the practicals that might come up tomorrow. It does tend to come up quite a bit in the biology paper 2s. Now, what the ruler drop test is, it's measuring reaction time. So if you want to test somebody's reaction speed, you could drop a ruler and just see how fast they're catching that or what height they're catching that and so on. So this is a mark scheme from describing how to do a ruler drop test for 3 marks. And this is what the mark scheme wants you to say. So hold a ruler just above the open hand of the other student. So ignore neither hand. They will not give you a mark for this. Meaning a lot of people put this down. So just be careful of that. So hold a ruler just above the open hand of the other student. You're going to drop the ruler and the other student's going to catch it. So do not give a verbal signal because it's nothing about a verbal signal. You just want to see when does the student actually catch the ruler. So that's your second mark. And then record where the ruler is caught. So ignore timing. A lot of people do measure the timing that they take to catch it, but it could be a millisecond. So how could you record that timing? So that's the reason why they do not want you to write about timing. They just want you to record where it's caught. So that's what the ruler drop is. Okay, so that's your 3 marks there. Explain why the ruler drop test does not involve reflex action. Well, what is a reflex action? A reflex action is something that you're just doing automatically without thinking about it. So just remember that. So a reflex action is something you're doing automatic and you're not thinking about it. But with the ruler drop test, you are thinking about it. It's not an automatic reaction. So that's what we're saying. So it's not automatic. So and it does not. So it's just explaining about the conscious part of the brain. Okay, that's that one. Okay, so moving on to the greenhouse gases. So this is relevant for your chemistry paper 2 exam as well on Friday. So for biology, they do emphasize carbon dioxide and methane being the greenhouse gases causing global warming. So methane tends to get like if they ask what's the cause of that, it's like farms. So more cows in a farm would cause more methane production. Carbon dioxide comes from stuff like combustion, burning fuels, and so on. So they're the greenhouse gases. You need to know about both about the carbon cycle and the water cycle. A lot more people struggle with the carbon cycle rather than the water cycle. The water cycle is just where waters are in oceans and rivers. They then can get evaporated into the clouds. They then condense and then precipitation, and then the cycle continues on and on. So that one's quite straightforward. The carbon cycle's a bit more trickier. There's a lot more going on. So this is another 6-mark question I pulled. Plants take in carbon dioxide from the atmosphere. The reason why plants take in carbon dioxide from the atmosphere is photosynthesis. So keep that in mind. Figure 2 shows part of the carbon cycle. So the arrows are just showing where it's going. So we can see carbon dioxide in the atmosphere. It's coming from the animals, because what do animals do? Animals do respiration. There's nothing wrong with annotating your paper as well. So just for you to understand it. So I can see from animals, they contribute carbon dioxide to the atmosphere because we're doing respiration, and respiration produces carbon dioxide. What else can we see here? Carbon dioxide from the atmosphere to the green plants, that's photosynthesis, as we discussed. Green plants to animals, well, we eat the green plants, and the carbon that's in the green plants, we're then taking that in. So that's how it goes to animals. Animals to microorganisms. When animals die, you bury them. What feeds on the animals? The microorganisms, in particular decomposers, are feeding on the animals. So the carbon's basically cycling around everywhere. And then you can see with the microorganisms, well, they're also going to be doing respiration as well. So that's why they contribute to carbon dioxide in that atmosphere. So it's quite nice that they've given you this diagram, but they don't necessarily have to give you this diagram in the exam. They could just give you this question and you're expected to know all this as well. So don't be reliant that they will give you this diagram to help you. Okay, so let's see where they actually give the 6 marks. So it says, describe how carbon from the atmosphere is cycled through living organisms. So everything that I've just spoken about. So let's see. They spoke about photosynthesis, and they've actually explained photosynthesis as well. So it's taken in through the stomata, used for photosynthesis to make glucose. So they've just, remember, you can even put the equation down. So I'll just write here. So that's why there's a lot less paper 2 content, because they still want you to know your paper 1 content for this one as well. So stuff like photosynthesis and respiration. So photosynthesis, the plants take in carbon dioxide and they react that with water that we give to the plant. They then release oxygen and glucose. So you could see that's explained here, the glucose part. The glucose is used to make other carbon compounds, such as proteins, lipids, and so on, and it's stored as starch. So you don't necessarily have to write everything, but they do want you to explain some things. Feeding: plants are eaten, consumed by animals, which uses carbon compounds to make other carbon compounds, as we just said over here. The green plants as well, again, eaten by the animals, carbon's transporting through. Decay: when plants, animals die, they're decomposed by microorganisms, the decomposers that we spoke about. They essentially are feeding off that animal that's buried. That's why it decomposes. It doesn't just stay as is. Okay, so you can see how there's one repeating thing in this. So which use the carbon compounds to make other carbon compounds. They're repeating the same thing for each thing as well. Even here as well, used to make other carbon compounds. And then finally, respiration. So plants and animals and microorganisms all respire, which release carbon dioxide back into the atmosphere. So I think where people go wrong is they just say just the first line. So they'll say photosynthesis, but then they're not explaining it. How's carbon actually cycling through? You have to give a bit more with these 6 markers. Okay, and then we've done that one as well. Okay, so another 6 marker here. So increasing number of cows are being farmed around the world. Explain the environmental implications of increasing number of cows being farmed. So this is all the methane effect and greenhouse effect that we were speaking about before. Whenever cows are mentioned, or any sort of like animals on a farm, it does relate back to methane, is what you should be thinking. Okay, so let's have a look at the mark scheme for 6 marks. So consequences of increasing the number of cows. So if you're increasing the number of cows, there's fewer plants due to more trampling, which makes sense. So if there's fewer plants, then there's less photosynthesis happening because they're not taking in the carbon dioxide. So fewer plants due to more trampling. So you have to explain it. Trampling from the cows. Due to more plants being eaten by cows. So less photosynthesis, as I've just described, so more carbon dioxide is in the atmosphere because it's not being used for photosynthesis. That's another reason why deforestation is also really bad as well. If you're taking these trees, which all photosynthesize, and a lot of them as well, you're still keeping a lot of carbon dioxide in the air because the trees are not going to use it for photosynthesis because they're not there. Okay, so that's one point. More cows will release carbon dioxide from respiration. Yes, all the cows are respiring, so they're releasing carbon dioxide. Cows also release methane, and if you've got more cows, more methane. Deforestation to provide farmland. So as we just spoke about as well, for these cows, if there's more cows that are going to be farmed, they need space to be farmed. So therefore, trees are taken down for this space to be there for the cows. More cow species entering rivers, streams, and watercourses. Okay, yeah, so it's saying explain the environmental implications. So I guess that's affecting the water as well. Okay, and then actually describing what's the issue with each of these. So carbon dioxide and methane being greenhouse gases, but why is it bad that they're greenhouse gases? Because of global warming, and so on. That's a good point actually there as well. So effects of deforestation, reduced biodiversity. If you're removing trees, which loads of species are reliant on, there's a lot of species that would be living in them trees. You're getting rid of all them species with deforestation. So therefore you're reducing the biodiversity. So the number of species that's there. Okay, let's cover some content in the last 15 minutes. Okay, so meiosis, we did not speak about. So for paper 2, you don't need to know about mitosis. It's meiosis you need to know about. So make sure you know what, let me try and copy this over. Okay, I'll just explain it rather than writing on it. So the first stage, so that first stage there, it starts off with a normal cell, so a cell that's got 23 pairs of chromosomes, or you can say 46. Remember, only say 23 pairs or 46 if it's a human. They might give you a different species and it'll be a different number. Refer back to the question then. Okay, so just like with mitosis, all the chromosomes need to duplicate. Everything needs to duplicate in the cell because it's going to get split again. So part 2, you can see the similar chromosomes pair up and the genes swap between them. The reason why the genes swap between them is because we want all the final cells to be genetically different from each other. And I'll come on to that why. So after part 2, when all the similar chromosomes have paired up and the genes have all swapped, the first divide happens. So at this first divide, we've got 2 diploid cells. So remember, each of them have 23 pairs of chromosomes, so 46. But we don't want 46, we need half the amount. So we do a further divide again. So one cell becomes 4 different cells, but each of these have 23, so they have half the amount of chromosomes. And that's because they're gametes. So the eggs in females or sperms in males. So let me just write that down here. So just remember, with meiosis, it starts off with 46. I'm going to say 46 rather than 23 pairs. That 46, so it'll duplicate, and then the similar chromosomes line up, and then it splits. That first split, there's still 46 because remember, they've duplicated, so it's a diploid, and it's still a diploid because everything's duplicated. But we don't want it to be a diploid. That's the process of mitosis. We don't want that. We want a further split. So they split down into 23 each because these are the gamete cells. So basically egg and sperm. If they ask you why is it half the amount, because when an embryo is formed, they fuse together to create the embryo. So 23 plus 23 should give you 46. So that's the reason why it has to be half the amount. Yeah, just be aware of the numbers when it comes to human chromosomes as well. So that's meiosis. And all of them are non-identical. So remember, with mitosis, they're all identical. Meiosis, they're non-identical. Every single one is different. If they were all the same, you would look the same as all your siblings. They have to be different. So we've got genetic variation between all the offspring. Okay, let me explain the difference between DNA. So gene, DNA, and chromosome. Technically, they're all the same thing. A gene is a section of the DNA which tells that DNA what it's going to do, what's it coding for exactly. Now the DNA, so deoxyribonucleic acid, is a really long strand of genetic information, and we can't have a really long strand of genetic information. So what happens is it coils up, and that's what forms your chromosomes. So your chromosomes essentially made up of DNA. It's just all coiled up and packed together, and then that sits inside the nucleus of a cell. So yeah, don't get confused. They're all the same thing. This is the order of size. So gene, DNA, and then chromosome. Okay, we've spoken about Punnett squares. We've talked about polydactyl and cystic fibrosis. So variations can happen genetically, or it could even happen to do with the environment. So for example, if you were to get a tattoo or you cut your hair, that's not all genetic. That's something you've chosen to do because of the environment. We've done selective breeding. Genetic engineering involves modifying the genome of an organism by introducing a gene from another organism to give a desired characteristic. So, for example, when you see GM crop, it's because they genetically modified crops. That's why they all look the same. So looking at that first example, plant crops have been genetically engineered to be resistant to diseases or to produce bigger, better fruits, or you just want all of them to look the same. So if you look at fruits that are just grown without being genetically modified, all of them tend to look different. But in supermarkets, everything tends to look the same. It's because they're genetically engineered to be like that. Okay, this is quite important as well. So the classification, I've not got a past exam question on this one, but they can give you like a species name, and you just need to remember this order. So kingdom, phylum, class, order, family, genus, and species. So you can remember it using this mnemonic on the right hand side. And that's just the order. They might say, what is this in so and so name? And you just keep counting on this classification method here. So make sure you remember that for tomorrow as well. Adaptations, that's quite an important one as well. So adaptations can be structural, behavioral, or functional. So for example, they would always give you the examples in the exam question, and then you just have to match what is it for exactly. So for example, a cactus has spikes on it, and that's an adaptation to make sure no predators eat it. What else could we say? For example, a polar bear has white fur, so it can blend in with the snow of where it lives, so therefore it's protected from predators as well. So it's always different on the context of the question. We've spoken about the carbon cycle, so, and then we've got the water cycle as well. That's there. Biodiversity, we've spoken about that. But yeah, that's everything from this session that I did want to cover. Has anyone got any questions or anything they want me to cover before I wrap it up there? There's a lot less content compared to all the paper ones for all biology paper 2, chemistry paper 2, and physics paper 2 for combined. Evolution. Okay, let's go over evolution. So the whole point of evolution, you can even think of it as the whole survival of the fittest thing as well, but then that leads on to natural selection. But the theory of evolution, that all of today's species have evolved from simple life forms that first started to develop, and then they were over generations, sorry, they were changing, and the ones that were adapted to essentially the survival conditions were the ones that survived, and their genes were passed on. So make sure you know Darwin's theory of evolution. So he says that random mutations result in variation. Mutation is just a change in the DNA, essentially, that's happened. Some organisms are better adapted to the environment. They compete, so survival of the fittest. Yeah, we'll send the notes today after as well. And those better adapted are more likely to survive, and over time, their characteristics are the ones that show in future generations. So they tend to look at today's generations, see what characteristics they've got, and then they can kind of predict what was there in the past as well that survived and basically passed on their genes. Also remember as well, when you're breeding two different species together, this is actually quite an important point. I saw this in a past paper. So when we're breeding, so where did I put it? Okay, so like we've just said here, when you get an egg and sperm, so 23 and 23, you get 46, so it works. If you end up breeding two different species, so for example, a tiger and a lion, so you get a liger, that liger is not fertile. So when I say not fertile, I mean that they cannot have babies, basically. And the reason why they can't is because their chromosome numbers do not match up. So say for example, they could give you any example in the exam, but let's say a lion has 11 pairs and a tiger has 8 pairs of chromosomes. If you then form a liger, so you've actually selectively breeded them together and to get their characteristics, this will not be fertile because it's got 11 from there and it's got 8 from there. So who is it going to mate with to then produce offspring? So just remember that if you combine 2 different species, they're not going to be fertile. I've seen that in a past exam paper as well. Okay, so that's what this point here is saying, the natural selection. I'm just finding where it was. So if 2 populations of one species become so different in phenotype, they can no longer interbreed to produce fertile offspring because they've formed then 2 different species. So that's the main point. 2 different species cannot breed together to form fertile offspring. But yeah, that's that. Anything else anybody wants me to go over? 64 out of 70 on a pass paper would be grade 8, grade 9 if you mark just that one paper. It's quite tricky to say because they don't do it on just that one paper. They give like an overall mark for it. So like if you're getting like an 8, 9 or 9, 9, it's just one mark over all 6 papers. So I guess you could divide that by 6 papers if you look at last year's grade boundaries, and that might give some indication. The negative feedback thing as well, make sure you're aware of how that works with stuff like thyroxine and TSH as well. So if your thyroxine goes too low, your TSH gets released from the pituitary gland. That tells your thyroxine to go back up again. But now if your thyroxine is high again, then your TSH level drops. So it's the same thing with the glucose and insulin stuff as well. It's still negative feedback. 289 out of 420. So if you do 289, or 289 divided by 6. So you need 48 in each paper, but a 9. Yeah, so I guess even though you're saying 64 or 70 on a past paper, would it be a grade 8, grade 9? It depends on what you get in every other paper, which is what's important. But anyways, I hope that was helpful. I'll get these notes sent out, and if you've got any questions as well, do ask. Do you think the boundaries might go up? I can't really say. Did they go up last year for science? I'm pretty sure they did, right? That's what makes it, I guess, less predictable. We don't know if, like, how everyone will do this year. It's quite tricky to say.